Hello, good morning, and welcome to the Hedge Fund Association webinar, Biotech in the Post-Pandemic World, Follow the Science. I'm Mitch Hackles, the president of the Hedge Fund Association. The HFA is a global nonprofit industry trade and nonpartisan lobbying organization with a mission to advance transparency, education, and trust in alternative investments. If you're not already an HFA member, we ask that you please consider joining, and you can learn more at hedgefundassoc.org. We will, of course, also follow up and send you information. Now, before we begin today's program, I'd like to thank our webinar sponsor, StoneX, for their kind sponsorship of this program, and HFA's annual Thought Leader sponsors, Citrin Cooperman and UMB Fund Services. We're also delighted to spotlight the Christopher and Dana Reeve Foundation as our charity partner. Now it's my pleasure to introduce Richard Westmark, the managing member and CIO of Triple V Management and HFA's Texas Regional Director. Please take it away, Richard.
Thank you very much, Mitch, and thanks everyone for joining the webinar. I'd like to make special thanks to Mitch and the leadership of the Hedge Fund Association, who I can tell you are making the Hedge Fund Association a dominant force in alternative investments. After our panel discussion, hopefully we'll have time and we'll open the audience up to questions. If you go to the GoToWebinar, that's where you can post your questions. You go in the control panel, you'll see a box where you can type in your questions, which we'll monitor and know about. Now let's meet the panelists, and thank you very much for our panelists for being here. The first is Nino Kioka. He's the guy in the white coat. Nino is chairman of neurosurgery at Harvard Medical School and a founder of multiple biotech companies, particularly leading the efforts of Harvard in their desire to build an ecosystem of startups. Joseph Lawyer, you can't see his picture. He currently runs a biotech fund. He founded and exited a gene sequencing company while he was in medical school. And this year he IPO'd Anabulo, which is on the NASDAQ, which is a biotech company that's in phase 2 trials for an antidote to marijuana toxicity. And Rex Marco, you can see there, is a world-renowned spine surgeon. After suffering a spinal cord injury, he's serving essentially as the chief medical investment officer for the Christopher and Dana Reeves Foundation, who we're proud to be supporting today. And they have the foundation has an effort to invest in mobility-related technologies, and Rex is leading that charge for them. My firm, Triple V Management, believes that superior investment returns can be realized through in-depth research rather than relying on sell-side analysts. In this light, I'm thrilled to host this rare opportunity to hear directly from these leading world authorities on pivotal biotechnology that's coming to market or in the market. I believe investors in both the public and private markets will derive actionable investment theses from their expertise, and I look forward to hearing from it. And Nino, it's all yours.
Thank you, Richard, and I appreciate the other members of the panel. Being at Harvard, we cannot speak without showing slides, and so I hope I can show my slides and tell you a little bit about what we've been doing here. I entitled this talk, "It's all about the RNA, stupid," because we're becoming a world where RNA is becoming a leading therapeutic. And really, this has been shown by the COVID vaccines, which showed the power of RNA technology in inducing immunity, and actually very potent and powerful immunity, more so than most of the other vaccines against this pandemic. And I hope you don't mind if I take you back to your biology classes from college, but sometimes when I speak to different audience, they ask me, what is actually RNA? RNA stands for ribonucleic acid. It's actually a chemical that's made in your body. But this chemical derives from DNA, and DNA, as you see here on the left, is the source code of life. the structure of DNA was discovered back in the 1950s by Watson and Crick. And then over the following 50 years, we had advanced the science all the way to sequencing all of the human genome, all of the human DNA. DNA is the equivalent of the software of your computer. It encodes all the information that basically makes you who you are, and it's then transmitted to your children and your sons and your daughters. However, DNA by itself needs that source, that software needs to be interpreted. So the interpretation is provided by messenger RNA. Messenger RNA is a circuitry of your computer that brings that software command from DNA to finally making what's called protein. So messenger RNA basically is the transmitter of that DNA information. And it turns into protein, and the proteins are the functional blocks. If we make this analogy with your computer, proteins is actually what you see on the screen. It's what makes the colors on your computer black, it makes you slides, allows you to see what the stock market is doing. That's what protein is. These are the functional blocks of your body. So we talked about vaccines, but there's been a lot of interest in understanding how DNA, RNA, and proteins are involved in cancer. And one of the problems with cancers, especially the most difficult cancers like pancreatic cancers or brain cancers, are still very impervious to treatments. And why is that so? So cancer is a disease where the software, that is your DNA, has become mutated. So it's like the software in your computer as a Trojan virus or as malware. And that makes your computer function funny or basically not function well at all. But when this happens to DNA, especially in particular genes or in particular pieces of software, it will make the cells in your organ, the pancreas, the brain, your ovaries, divide continuously. And in this process of division, continuous division, form a cancer or tumor, which can then metastasize throughout your body. Now, there's been a lot of interest to understand what these software changes are, where are the mutations, and then devise targeted therapies, such as TKI, which are tyrosine kinase inhibitors, for example, against the mutant proteins that are coded by these mutant DNA genes. And this has been good for some cancers, but for some of the more difficult cancers that are listed here, the problem is that those cancer cells are so heterogeneous. In other words, that target is not expressed in all of the cancer cells, that when you target only some of the cancer cells, all you're doing is you're making the other cancer cells that do not have that target grow better. And that has been a problem with targeted therapies. So people have turned to immunotherapies. In other words, get your own body immune cells to reject the cancer. The problem is that a lot of these cancers have also evolved to be able to actually fight against your body's immune cells and actually even educate your body's immune cells to become tumor promoting. In other words, making your body immune cells help the cancer grow rather than fighting against it. And that is the difficult thing with some of these most difficult cancers. And so the question that we have raised and others, can we use this power of RNA technology to fight cancer? So this is not novel. And so this is basically the same technology that is being used for the vaccines, which is once you know what an antigen would be the what your immune cells recognize it. So for example, for coronavirus, the antigen is the so-called spike protein. Once you know that, you can make an RNA molecule that codes for that antigen, and then have some type of delivery method to express it into your immune cells. So your immune cells will now recognize that foreign protein, that antigen, and attack it. So people have wanted to do this for cancer for a long time. Identify what those mutant proteins are, and then make either DNA or protein. But now we know that if you actually use RNA, that's actually the best system. And so this is companies like Moderna or Pfizer, Biotech, their initial goal was to do this. They were not made to, or they were not started to go against coronavirus. They were started to try to make this type of technology available for cancer treatment. And as you can see, this is from 2015, way before coronavirus happened. So now I'm gonna tell you one more thing, which is when we talk about RNA is the transmitter information, we're really talking about messenger RNA. This is the message that goes from DNA to protein, okay? It turns out that there are a number of other categories of RNA, which we used to not know what they did. It's called non-coding RNA. It just does not make protein. That does not have the ability to make protein, but it's there, and actually there's a lot of it in the cell. And it has all sorts of names based on how big it is, based on their structure, such as long non-coding RNA, circular RNA, microRNA, and so on and so on. And it turns out that when you look at the entirety of the human genome, all 4 billion bases of it, and so if you look at all the software, only 1% of that software shown in this little sliver is made of messenger RNA that's actually encoding for proteins. So it's a really tiny sliver. Everything else is encoding for RNA that does not make protein. Well, what is this? Why has the body, why have humans evolved to have all this stuff that's not making a building block? Well, it turns out that this is the regulatory part. So as I said before, the central dogma we call is that DNA makes RNA, which then transmits the information to make protein. But it's not that simple. What actually happens, you need to have regulation of this. You need to regulate when will the DNA make the RNA, when will the RNA make protein, and what time of your life cycle. Let's say if you're an embryo, a fetus, a young adult, an old adult, in what organs, is it the brain, is the pancreas? And one of the major regulations of this is coming from all these non-coding RNAs. These non-coding RNAs actually are what regulate when DNA goes into RNA and when RNA becomes protein. So it turns out that over the last decade or so, this has been recognized as an important component of cancer. As I said, cancer is a disease of mutant DNA genes that end up making mutant messenger RNAs that make mutant proteins that become basically the cancer-producing agents. I'm just trying to remove this from here. So we work here. We work on these non-coding RNAs. We want to understand this regulation. And we've been trying, and we are working on commercializing 3 technologies that are covered by intellectual property from my place of work, which is called Mass General Brigham. And these 3 technologies are trying to address 3 features that are common to all cancers. The first feature is all cancers know how to get rid of your immune cells, know how to evade and escape your immune system's ability to kill off any pathogen or any abnormal tissue. The second is, as I said, cancer cells develop what's called pathological stemness, which means self-perpetuation. They're able to continuously divide, forming these cancers. And lastly, is that they're able to resist therapy, like radiation, chemotherapy, surgery. They find a way to basically live through all these things that we do to patients and continue to grow as a cancer. So our technologies are trying to address one at a time each one of these features. And we believe and we know that non-coding RNAs are the key factors in the 3 features of cancer. So I'm just going to give you the 3 examples. So this is a non-coding RNA. It's a specific molecule that we discovered. We gave it a name, INCAR1. As scientists, we always like to give very hard, difficult names. But basically what this non-coding RNA does is allows your tumor cells to become, to basically hide from your immune system. And we discovered that if we remove this non-coding RNA, your immune system now recognizes the tumor cells and rejects them. And this is just an experiment, and I'm going to try to explain this. So these clumps of green things are called tumor organoids. The green means that these tumors are very much alive, they're growing. What we do now is we're going to remove this non-coding RNA, and when we remove the non-coding RNA and add some tumor killing T cells, or these are tumor killers that come from the body, what you can see is that when you remove this non-coding RNA, these cells are not green anymore, they're dying off. And so basically these T cells now are able to act more efficiently in killing off these tumors. I'm going to give you an example about the second part, which is tumors learn how to self-perpetuate, they learn how to grow. And one of the things we've discovered is one of the reasons they can do that is they're able to remove all the RNAs that have something to do with suppressing growth. And we found a mechanism that happens, and I don't want to go too much into detail here, but basically there's a particular factor in the cell that allows RNAs to basically stop the tumor from growing. And what we found is that in tumor cells, there's a particular non-coding RNA called Circ2082 that blocks this factor. And when it does this, basically you don't have any more of these tumor suppressor messages being made. And that allows the tumor to keep on perpetuating itself and growing. And the last one is involving resistance to therapy. And one of the things we've developed is these chassis, these frameworks of chemical nucleic acids, where you can plug and play different microRNAs or RNAs that basically block tumor cells from growing. And so we have these constructs that we make and where we can deliver multiple RNA molecules. And here in this case, for example, this is a tumor in a brain, this is a mouse. And if you add just 1 microRNA, these tumors grow less. At 3, they don't grow. And if you keep on adding to this, you basically make these tumors completely disappear. So we're building platform technology biotech that is really focused on the power of these non-coding RNAs. These are the RNAs that regulate that process from DNA to message to protein to fight the 3 main features of cancer. And with that, thank you and I appreciate any questions. And this is my email if you wanted to email me questions.
Nino, I have a question for you.
Yes.
And for the people listening, I want to summarize what you've just said, which is you're talking about advances that are going to cure cancer. Like really, truly, the technology is getting good enough that we're on the cusp of not just treating cancer, but curing cancer. And to the investors watching, that's such an amazing thing that this is the nascent period of being able to cure cancer. Specifically, Nino, the technology of Pfizer and Moderna, the mRNA technology, the vaccine technology, do you see that as translational into cancer therapy? That is, if we're betting on companies, are those 2 good companies to bet on for this, or are there other companies out there that you think are maybe ahead of them?
So first of all, I don't give investment advice, so that's for you. The Moderna and Pfizer technology was actually initiated to go against cancer, not coronavirus. So basically what they want to do is find the tumor mutant peptides from each patient and then design messenger RNAs that coded for those peptides and then basically inject them into your arm so you could be vaccinated against those tumors. So that's novel, revolutionary. We will have to see how that works against cancer. If I had to criticize it as a scientist, but again, it's a scientific criticism, the concern is that you basically have to design for each patient and for each cancer a novel messenger RNA cocktail because all the cancers are different from each other. Coronavirus is just one, but you can already see as the variants and mutants happen, we're not talking about making different vaccines. So think about that with cancer, where you'll have multiple of those antigens that you have to go up against. So that's one problem. And the second problem is going to be that it's really something that's very downstream of our cancer forms. I think the non-coding RNAs I'm talking about is an upstream. It's a vulnerability. It's something that's making those cancers work. And if you eliminate that command and control system, you eliminate the entire cancer. That's why I think it's exciting. And so I think that any company that's studying the regulators more than the actual final product is an exciting company to look at. And I cannot give you names.
Yeah, no, thank you very much for that. And thank you for that talk. And I take it that that's the contact that you would ask for if anyone wanted to talk more about that technology.
Yes.
All right, well, thank you again. Joe.
Yes, Richard.
You ready to go?
I'm ready to go. So I don't have any slides, so the absence of video shouldn't be limiting. Richard asked me to speak with you today about the newest, shiniest object I see in biopharma. And before I do that, I should mention, by way of disclosure, that I'm a short seller, which means I see many things that are shiny on the outside, but rotting within. So my enthusiasm will be tempered by the many previous shiny objects I've seen that have not lived up to the expectations placed upon them. This talk is relatively high level, but I'm happy to go into details if anyone wants. So let's set the stage for where we are presently. In some ways, we're the victim of our own success. We, and that is big picture we, the physician community, do a tremendous job with the pharmacologic management of common diseases such as hypertension, hyperlipidemia, depression, rheumatoid arthritis, and the like. We don't do as well regarding the treatment of cancer, but there's been a steady wave of drugs that increase survival, just not to the extent we would like. And we have a very long way to go regarding many neurologic diseases where, frankly, we haven't a clue to the underlying biology. But I think what the offshoot of this is that we're really running out of diseases that affect a large number of people where we know enough about the disease biology to develop a drug. And to be clear, this is a good thing. So if we think about improving human health, we have to consider the question from two perspectives. The first, and far more difficult problem, is having a small but meaningful impact on a disease that affects many people. For example, we will all suffer from diseases of aging at some point, but we really don't have any idea how or why we age, which has made the diseases of aging quite difficult to develop treatments for. It's a hard problem. So difficult, in fact, that I think the best way to improve health outcomes for the largest number of people is to reduce harm, self-inflicted largely harms, such as smoking, obesity, and the like. There are other highly prevalent conditions that we might impact on, and I'll have more to say about this later. The second and easier problem is making a large impact on the disease that affects relatively few people. It turns out we're good at this. This has been a trend in medicine for several years now. Companies develop orphan drugs and charge incredibly high prices for them in order to make the economics of doing so viable, but these drugs frequently do make a large impact on the disease. In my opinion, addressing this group of less common diseases with genome editing using CRISPR is the most exciting technology in healthcare right now. Let's step back for a second and just review what CRISPR is and what it does. CRISPR is a surveillance system developed by certain bacteria as a defense against virus infections. It works by being targeted to and cutting a specific sequence of DNA. In the case of bacteria, this certain DNA is that of a virus invader. What researchers have been able to do, though, is adapt the system such that it can be delivered to human cells and affect specific genetic alterations within them. Now, let's distinguish gene editing by the place where it happens, because I think that's a useful way to think about the technology. We have ex vivo editing, where the editing is being performed in cells outside of the body. This has been done for a while now. You've heard about CAR T therapy for cancer, which is one form of ex vivo gene editing. But there are several different ways of doing it, and CRISPR is just one of many technologies that you can use to do this outside the body. In fact, as a brief digression, embryo editing is a form of ex vivo editing, and while we may not permit it in the United States, it'll probably happen in other nations. It made headlines some time ago when a Chinese scientist edited an embryo and then implanted the embryo in a woman who delivered a baby to term, and that baby was in fact edited for the trait the investigator sought to edit, although I'd add that someone with the intelligence to do this experiment should also have had the intelligence not to do the experiment. Well, anyway, after that scary thought, let's turn our attention to in vivo editing, where the editing is occurring in the patient. It's different because the DNA is being edited in the person in a living cell. So you really have to get it right because there's no chance to do quality control outside the person before you introduce the cells. For example, if a CAR T manufacturing process goes wrong, you can start again. But if you're editing in vivo, you don't have that option. So a few months ago, Intellia demonstrated for the first time earlier this year that they could edit the gene responsible for TTR amyloidosis in a patient. This is a rare disease and one for which we already have treatments, both small molecule and RNAi-based approaches, but still it's an excellent proof of concept for the technology. The key difference between the RNAi-based approach, in which a patient receives a shot every 3 or 6 months to suppress expression of the gene, and gene editing, is that the gene editing is permanent. As far as efficacy goes, they both appear to be the same, although the RNAi-based approach, by virtue of being around longer, has much more safety data. Now, there are risks with CRISPR, such as off-target editing, which is basically editing the wrong part of the genome. And should this happen, it's irreversible. This off-target editing could be a silent mutation, in which case we would never know about it, or it could be a cancer-causing one. We simply won't know how often this happens, if at all, until many more patients are exposed to the technology. The other point worth making is that the approach used by Intellia is not heritable. So the person who is cured still has the same odds of passing the disease on to their children, which raises an interesting semantic question about the use of the word cure, since if you can pass the disease on, are you truly cured? But in any case, why is this a big deal? Well, the reason is because we're now talking for the first time about delivering cures for certain liver diseases. And if the technology develops to deliver the editing machinery to specific cell types, such as the kidney or the eye, we'll be able to cure additional diseases in those tissues as well. This is important because the more diseases we can reliably call cured, the better off we'll be. And over time, as these approaches lose patent protection, the cost of treating many of these diseases should come down quite a bit. Which brings me to the dilemma that investors are presented with, because there simply aren't a lot of rare diseases to support so many multi-billion-dollar market capitalizations, right? They're rare for a reason. If CRISPR catches on, then you'd think that the RNAi-based approach taken by companies like Alnylam and Arrowhead and Dicerna will fall out of favor, and presumably those companies will suffer share price declines as a result. If, on the other hand, CRISPR has a safety setback or the data, for some reason doesn't pan out, then the RNAi-based companies will benefit and the editing companies will suffer. It's difficult to believe that there will be a Lake Wobegon effect where everyone's above average. In other words, both technologies win. So this sector is an interesting place for making pair trades. Now, let's return to the idea of making a small but meaningful impact on conditions that affect many people. I think addiction is one of those conditions. And when I initially thought about drug development for this condition, I assumed that battling the receptor pharmacology would solve the problem. This works for the short-term treatment of overdoses, and Narcan is used quite successfully to treat those overdoses. When I learned how many people were showing up at emergency departments across the country with what's called acute cannabis intoxication, I started a company called Nebula to develop an antidote. And as Richard mentioned, we just went public and hope to have phase 2 data next year. But when we think about drugs of abuse in this country, there's going to be a lot of people who want to quit but need help doing so. In a lot of ways, it's like obesity. And if one can successfully develop a drug in this indication, they won't have to price it at a very high level to have a nice business because there are so many patients who would need it. And ideally, it would be something that one doesn't have to take forever, but rather some prescribed period of time after which they would be able to manage their addiction without it. So anyway, we've spoken about drug development from both sides of the spectrum. We've talked about developing cures for rare diseases with gene editing on one hand, and developing drugs to treat drug intoxication and drug addiction on the other. In both cases, we have a good enough handle on the biology that we can aim the tools of medicinal chemistry and biotechnology at the problem with some reasonable expectation of success. But investors will not support drug development without an expectation of a return, which brings me to the final point. From an economic perspective, biotech's a terrible industry. All of the easy drug targets have been discovered. If you think you have a drug target, you'll spend tens, if not hundreds of millions of dollars proving out your hypothesis. The majority of these programs will end in failure, despite an FDA that's the most permissive in history. If you do secure an approval, you'll most likely be dependent on the ability to price gouge in order to make a profit, and even then, it isn't clear. I mean, just today, there was an announcement by the White House about an effort to rein in drug pricing. So your drug will almost certainly have competition after your patents expire, and generics will enter the market and your sales will go away, and then you have to do it all over again. If you look at the accumulated deficits of biotech companies that we would all call successful, you'll see that in many cases those numbers are negative. So you might ask, well, why people do this? Why do people do this? And the reason is because every now and then you find a drug like a Keytruda or an ibrutinib or a Kalydeco, which provide meaningful benefits to patients and investors alike. And so even for drugs like these, home runs by any standard, there are problems because many times these companies have to recoup the bulk of their development costs based on the sales in the United States. And that's because we have a lot of regulatory dysfunction in the United States when it comes to drug pricing. So we've essentially subsidized drug development for the rest of the world. And while Americans celebrated Thanksgiving last year, the Chinese government established payment levels for various prescription drugs. The average discount in price relative to what these products sold for in the United States was 61%. And this is a direct wealth transfer in the form of subsidized research and development. And it's not just China, it's Europe as well. Americans are effectively paying through higher drug prices for the innovation that's sold around the world at substantially lower prices. So because drug costs in the United States are routinely higher than the rest of the world, they're an interesting example. They will not go away unless there's a political response, because unlike the United States, which functions as a pseudo-free market when it comes to drug regulation, in other countries there's a single buyer, it's single payer, and so they have a lot more pricing power. So what might we do here to fix that? And I think the answer is to effectively export our drug price inflation. And the way we can do that is through a few steps. The first is we can eliminate the medical loss ratio, which is a feature of the Affordable Care Act that requires many insurance companies to spend at least 80 or 85% of the premium dollars they collect on medical care. If an insurer fails to meet the applicable MLR standard in any given year, the insurer is required to provide a rebate to its customers. And my guess is that the individuals on this webinar haven't received a check from their health insurance lately, which tells you that they are getting right up to their MLR every year. But the practical impact of this is that health insurance companies can only increase profitability when costs rise. And that creates a feed-forward spiral of rising costs met by rising premiums. The second thing we could do is to allow drug importation. There's no reason to believe drugs sold in G20 nations are any less safe than those sold here, especially in light of the fact that the active pharmaceutical ingredient is often manufactured in the same factory. Those unpersuaded by this argument can continue to buy American, albeit at a greater cost. Now, the companies that make these drugs would respond to this by limiting exports to block their reimportation, which is why we'd need to couple drug importation with the most favored nation status amongst the G20 for government purchasers of drugs. This would end the US subsidy of foreign healthcare. And drug companies could continue to set whatever price they wished, but no longer could they charge Americans more for drugs that they sell abroad for far less. Finally, I would say that there are two other things we could do. We can make as many drugs available over the counter as possible. When I've traveled overseas, I often ask at a local pharmacy which drugs are available that require prescriptions and which ones are available over the counter. And in almost all the other countries I've traveled to, medications for chronic use are available without a prescription. And by that I mean drugs like antihypertensives, statins for cholesterol, anti-diabetes medications, contacts, in many countries oral contraceptives, hearing aids, and the like, are all sold over the counter. And the cost of these things would drop considerably if that was allowed to happen, because there would be that many more competitors. And let's face it, they're hardly drugs of abuse. Finally, we can insist before approving any drug that it be studied clinically against the current standard of care so that its value to the treatment armamentarium can be objectively assessed. European regulators often require this, while the FDA rarely does. I know this has been a wide-ranging talk, and so let me, in the remaining time I have, summarize. First, it's easier to make a large improvement in a rare disease than a modest improvement in a common disease, and this is largely because the low-hanging fruit has been picked. CRISPR-based technologies promise cure for many diseases, but they're going to compete with RNAi-based technologies. When it comes to chronic diseases for which we can probably make a difference in outcomes, addiction is one that's right up there. And finally, we can get a handle on drug costs in the United States. It can be done, but it's going to require an active political will that thus far hasn't happened. So I'll stop there and ask if you have any questions.
Hey, Joe, we're short on time, but thank you for that. I do have a question that I think is important, which is that given this price disparity internationally, it almost seems like the big pharma, if you will, companies are going to find huge margin pressures. Do you agree with that or not?
Well, I think they will if the political will can be gathered to affect some of these changes, right? I mean, the reason we have a drug price problem in the United States is precisely because the free market is not allowed to act. So even though there have been many proposals under President Trump and now under President Biden to rein in drug costs, the lobbying organizations have effectively beaten them back at every turn. So if something passes, yes, they will come under tremendous margin pressure. But thus far, it's not clear that they'll pass.
All right. Well, thank you, Rex. I think you're up.
Well, thank you so much for this opportunity, Richard and Mitch. I'd like to begin with my story and then talk to you about private companies and public companies with some exciting research for spinal cord injury as well as spine surgery, and then some industry trends as well as how to stay current in this market. Next slide. So I'm a surgeon and I'm a spine surgeon, and my main focus was on treating spinal cord injury or preventing spinal cord injury or severe deformity. I also had a passion for snowboarding and mountain biking as well as coaching baseball. On July 21st, 2019, I turned to the left instead of the right while mountain biking, and I heard a crack as I flew over the handlebars. And soon I realized that I was injured and I couldn't move my arms or legs, and I was paralyzed from the neck down. And in my recovery in the ICU, I had visions of working for the Reeve Foundation to help increase spinal cord injury awareness and find treatments and cures for spinal cord injury. And through this foundation and other connections, I realized that electrical stimulation and stem cells are the way of the future to help us find treatments for spinal cord injury as well as potential cures. And epidural stimulation can work in a couple ways. The first one at the top is implanting the stimulator on the nerve sac, either at the nerve root level or the spinal cord level. And I have a friend who's 38 years paralyzed from the neck down, and now he's moving his legs voluntarily and standing with minimal assistance through an implantable spinal cord stimulator. And then really exciting technology is placing electrical stimulation pads on the back of the neck and allowing patients to have improved arm movement with that. And this one company called Onward is a private company, and the private companies throughout this talk will have 2 asterisks by them. And they've raised millions of dollars and have FDA clearance to study the implantation and skin application of spinal cord stimulators. And in fact, the Ree Foundation and other not-for-profit foundations are partners with this company Onward. Next slide. The electrical stimulation projects are also being supported at major universities like the University of Louisville in partnership with Medtronic, which is a large company, public company, and they've received an NIH grant of $7.8 million to help support this research. Next slide. Another exciting opportunity for investors as well as patients and physicians is stem cell therapy. And this stem cell treatment by Dr. Biden on the left and his patient on the right, who was paralyzed while surfing, he was a super responder, and he received stem cells derived from his abdominal wall, fat cells, and then injected into the nerve sac. And he's a super responder, and he's been able to walk and move his arms since that treatment. Next slide. A promising private company started by the world-renowned neurosurgeon Michael Failings is Intelligex, and they're funded by the Toronto Brain Institute as well as the University of Toronto as startup funding for stem cells derived from neural stem cells and used to treat spinal cord injury. And this is a really promising technology to cure spinal cord injury. Next slide. Spinal fusion is an important aspect of spine surgery, and 2 companies that are have really promising technology in the fusion industry are Kuros as well as Therapeutics. And Kuros is using a hormone called parathyroid hormone, or PTH, combined with fibrin, which is the glue for healing, the start of healing. And that combination, I think, is really promising and is really warranted to keep an eye on as their studies go on and further fusion studies are obtained. And Therapeutics is using a peptide, which is a small portion of a protein, in combination with the calcium phosphate minerals that make up bone, to bring in bone forming cells and form what looks like near normal bone between the 2 vertebral bodies. So Kuros and Therapeutics are 2 private companies that are to keep an eye on in the future. Next slide. Navigation has become a really important part of spine surgery for many spine surgeons. And just like in your car, many people now require navigation to get from place to place. And some people actually require navigation to get from work to home and home to work. And likewise, in spine surgery, many surgeons are using navigation nowadays in almost every case, if not every case. And I predict in the next 10 to 20 years that at least half the cases in America will be navigated. Clearly, the leader in this industry is Medtronic, and they're pictured in the upper left. And that device is an intraoperative CT scan, and all the monitors are components for the navigation. And the next slide over is the next company that is really done a good job of coming up on their heels, and that's Globus with their navigation technology. And the next company is Nuvasive, and they are coming out with a strong navigation system. One of the new upcoming navigation systems is 7D, which was recently acquired by the public company C-Spine, and that was a $110 million acquisition this year. And that navigation system is really promising and very different than the other navigation systems that we've had in the past. And another private company involved in navigation is BrainLab, and they're partnered with Johnson & Johnson, and those two are a powerful combination. Keeping an eye on that partnership is important for investors, as BrainLab at some point may be acquired by one of the major companies. Next slide. So artificial intelligence is a very important part of spine surgery moving forward. And the leader in artificial intelligence right now is Medtronic with their acquisition of Medicrea at $158 million. That acquisition allows them to combine artificial intelligence with navigation as well as robotics, and that helps them work with surgeons to recommend optimal alignment with research and information that they wouldn't otherwise be able to easily put into their armamentarium to get better outcomes for patients. And also, what it allows them to do is to preoperatively bend a rod that can be placed more easily in patients. And that combination of artificial intelligence and navigation is now being combined by Medtronic with robotics as well. And Medtronic's acquisition of the Mazor company at $1.7 billion 2 years ago was a big acquisition and places Medtronic at the forefront of navigation, artificial intelligence, and robotics in spine surgery. Admittedly, Medtronic says that robotics is less than 10% of the industry right now for them. But moving forward, I predict that at least 25% of spine cases in the future will be with robotics. And other people in the market to bring robotics to the industry are Globus, which already has a robotics implant or instrumentation right now, and Nuvasive and Stryker will likely have the software to bring robotics to spine surgery in the upcoming years. And BrainLab also has a current robotic system that I believe can be used for spine surgery. Next slide. One of the really promising technologies to keep an eye on is augmented reality. And as you can see, the spine surgeon here now has the ability to be looking at the spine and seeing the three-dimensional anatomy of the spine as they're doing the surgery and putting in the screws. And this is really important as this technology improves and the safety and accuracy and live data improve. This technology, I believe, is the wave of the future and will really help patients have a safer, more accurate type of spine surgery. And Augmentex is the private company that's leading this type of technology. And as you can see, they were able to raise $36 million recently with this technology. Next slide. Personalized 3D printing is really intriguing, and as you can see, we can get these beautiful models of patients with tumors seen in purple on the spinal column with the aorta, which is the large blood vessel from the heart, and all the pulmonary tree and the skeleton all combined. Though not practical from a business standpoint or for every patient, the next slide shows you a private company using personalized 3D printing to build specific implants for patients with spinal deformity. As you can see, the rainbow colored spine shows a scoliosis curve that's corrected with a personalized interbody cage that is made to help correct the deformity. And Carlsmed is this private company that recently partnered with Precisive Surgery to help, or Precisive Surgical, to help improve outcomes with preoperative planning and artificial intelligence combined with personalized 3D printing. Next slide. Now, staying current with this technology and the fast-moving spine industry, one of the more promising companies that's run by my friend and colleague Jeff Rowe is Intuitivex. And they have more than a dozen portfolio companies and have raised more than $50 million as well as created value for their shareholders by following the technology and taking companies from inception all the way to commercialization. Next slide. And some of these companies are Precisive Surgical, which focuses on artificial intelligence and preoperative planning, and NavLab, which is a company focusing on artificial intelligence and augmented reality, and chiral surgery or surgical, which devises and makes implants that are favorable for left-handed surgeons, and Nova approach, which is an approach through the belly button rather than making other incisions for patients with spine surgery, and Amplify, which is an outpatient surgical company that focuses on improving the outpatient outcomes and less invasive surgical procedures like endoscopic spinal surgery, which I believe is going to have a major role in the future. And so Intuitivex is looking at endoscopy, artificial intelligence, augmented reality, as well as navigation and robotics. And as you can see, the spine industry is a $10 billion market. And to me, it's really interesting because 20 years ago when I started, Medtronic and DePuy and Synthes were the major companies and pretty much the only players. And now 60% of the industry is from other companies, including newer companies like NuVasive and Globus, ATEC, and C-Spine. And you can see that these are the major players now, Medtronic, Johnson & Johnson, and Stryker. And I don't know how to read these, but to me it looks like they've performed well. Next slide. Other public companies that are doing well and should be watched are Globus, C-Spine, Alphatec, and I'm not sure why NuVasive and Kuros are red, but to me they are promising and are leaders in their field. Next slide. Next slide. So in conclusion, I think navigation and robotics are here to stay, and the next technologies favor a tremendous benefit for the patients, including electrical stimulation, fusion products, artificial intelligence, augmented reality, and endoscopic surgery. And these will benefit patients as well as investors, I believe. And hopeful technology includes stem cell therapy as well as gene therapy for spinal cord injury. Next slide. So this is my contact information, and we're having an upcoming event for the Reeve Foundation to increase spinal cord injury awareness, and that's the Reeve Run and Roll. And I encourage you to join my team to increase spinal cord injury awareness. Thank you.
Rex, that's fantastic. Thank you very much.
This is a list of private companies that I talked about. I did not put Kuros on there, but that was just a mistake because Kuros is promising technology as well.
Well, thank you again. Thank you to all the outstanding panel. It's biotechnology. You can see the spectrum from proven public companies to kind of not sure of private companies, but more importantly, it's a growing field. I thank you all again. We will send you a follow-up email, which will include a replay of the webinar and ways to get in touch with the HFA, and have a great afternoon. Thank you again.
Thank you, everyone.